It's my pleasure to present the preliminary results of an ongoing trial, the MyPathway trial. That's a open-label phase 2 study looking at several molecularly targeted treatments in patients with advanced cancer. An increasing number of targeted agents for advanced cancer are in use now based on the presence of molecular abnormalities in the cancers. Examples of major successes in this area include HER2-targeted treatment for HER2-positive breast cancer and BRAF-targeted treatment for BRAF-mutated melanoma. We've known, though, that the same mutations that are in those cancers are found in a wide variety of other cancers, although at a lower incidence. And it's been difficult to test how effective these same treatments are in the other cancers due to the difficulty in identifying the patient population. With an increase in comprehensive genomic profiling that's gone on in the last few years, we're now identifying more and more of these unusual mutations in other cancers, and that's what this trial is addressing. There are 4 targets that are being addressed in this trial: HER2, BRAF, Hedgehog, and EGFR. The 4 corresponding treatments shown in the second line there on the slide are all drugs that are and treatments that are in use already and are approved for at least one type of cancer. Now, patients that are eligible for the MyPathway trial have non-indicated cancer types that contain one of these mutations. These patients also have had to be refractory to other standard treatment before being considered for this trial. This is a summary of all the patients that have been accrued and treated so far, 129 patients, and you can see in those patients, 29 actually have had a major response defined as a shrinkage of the cancer by at least 30%. Responses have been seen with all 4 of the treatments in this trial. No new safety signals were observed, and it's a little early to know how long the responses are, but you can see there on the slide that they're going to be in the range north of 6 months and probably between 6 and 9 months as we get more follow-up on this study. 2/3 of the patients had HER2 abnormalities, and on this slide, the largest group, those with HER2 amplification or overexpression, is shown. 61 patients in this group, and you can see on the bottom line, the overall response rate in this group was 28%. 6 different tumor types had responses: colorectal, bladder, biliary, non-small cell lung cancer, pancreas, and head and neck cancer. Of particular interest are the top 3 lines, the biggest groups right now. Colon cancers, these are refractory patients with a median of 4 previous regimens. 7 of 20, or 35%, had responses. An additional 3 had stable disease for more than 4 months, for a total clinical benefit rate of 50%. I think that's a very strong signal there. Although smaller, the signal is also strong in bladder and biliary cancer, each with 3 responses in those groups as well. Targeting BRAF in these patients was also successful. You can see of 33 patients, we had 8 responders, or 24%. In this group also, there were 6 different cancer types that responded to treatment: non-small cell lung cancer, ovarian, unknown primary, colorectal, pancreas, and head and neck. The only sizable group here was the non-small cell lung cancer, where 3 of 15 patients, or 20%, had responses, and another 2 had stable disease. And now, of interest in this group, 7 of the 8 responses were in V600E mutations. And as you know, that's the mutation that's been specifically correlated with high response to BRAF inhibition in melanoma, where this treatment is now approved. So looking at only those patients, the response rate here was 38% in this group. So in conclusion, I think we've shown now that this trial design is feasible, where patients are selected on the basis of molecular abnormalities in their cancers rather than on their primary tumor type or primary site, and certainly offers opportunities for patients with these molecular abnormalities. We saw responses in 12 different tumor types already in this trial, and in 4 major categories, including HER2-amplified colorectal cancer, bladder cancer, and biliary cancer, as well as BRAF-mutated non-small cell lung cancer. All of these groups met the protocol criteria for efficacy and have been expanded. And MyPathway trial is continuing to accrue patients, and just this last week went over 200 patients accrued so far. Thank you.